is Sydney Ashenbrenner, and I did abundance, vertical distribution, and vegetation preferences of salt marsh snails in the 312 salt marsh. My study site is across from South Street and Washington Street, here in the 312 salt marsh. Abundance, vertical distribution, and vegetative preferences of salt marsh snails. My hypothesis is salt marsh snail dispersion is random with no specific preferences in vertical location and vegetation, and species abundance is equal across the study site. Polygyrocereolus is the common name of the southern flatcoil. The shell is shaped like a disc and the foot is yellow. The shell is white with streaks of gray or brown. It has 7 to 9 whorls and is 3.5 to 4.6 millimeters. It has no gills and a paleo lung to breathe air. The shell top may be flat or pointy. The southern flatcoil consumes clover and alfalfa and is native to Florida. Polygyrocereolus has a phallus that protrudes between the two optic tentacles. Melampus coffea is commonly known as the coffee bean snail. The shell is ovate or teardrop shaped. The coffee bean snail migrates vertically as the tide rises. This allows them to escape predation in water. Snails have a brown color, lighter colored bands, and can be gray, tan, or yellow. The aperture is long and thin. This snail does not have an operculum, which is the door to close the snail in. It lives in intertidal areas along the coast of Florida and the Caribbean. The coffee bean snail will live around red, black, and white mangroves. They do not have gills and breathe through a hole in their mantle cavity. The coffee bean snail eats detritus and mangrove leaf litter. The coffee bean snail is preyed on by white ibis. Literaria aerata is commonly known as the marsh periwinkle. Marsh periwinkle's preferred habitat is Spartina and Juncus. Fossils suggest that in a post-glacial period, Literaria aerata expanded up to New England. This snail lives in brackish or salt water. The shell is ovate and conical. It has 5 whorls and a pointed spire. Adults can be 20 to 32 millimeters. The outside of this snail is gray-white. It can often have a slight amount of reddish-brown. This species is native to the Gulf of Mexico and Texas. Marsh periwinkles do not live in South Florida. Literaria aerata females are larger than males and lay eggs. Marsh periwinkles consume isomocyte fungi that grow inside wounds created by their radula. Snail defecation fertilizes this fungus. Literaria angulifera is commonly known as the mangrove periwinkle. The shell can be bluish-white, orange, yellow, red-brown, gray-brown, or gray-white. The mangrove periwinkle often has stripes or spots and 6 whorls. At max, the mangrove periwinkle can be 3.2 centimeters. The shape is elongate conic, like a long oval with a point. The mangrove periwinkle has a range that extends from Florida to Brazil. Literaria angulifera is ovoviparous, which means their eggs hatch inside their bodies and are released as larvae. The mangrove periwinkle grazes on algae and fungi. Some snails have a home site which benefits them more than foraging in the substrate and protects them from predators. The shade of the mangrove trees keeps Literaria angulifera from desiccation. For my methods, I walked haphazardly along the shoreline in waders from one end of the study site to the other. Some zones in the marsh were too muddy to cross, but I did try to sample 3 main zones: shaded Spartina, sunny Spartina, and shore of mangrove trees. I sampled 2,454 snails. In the coffee bean snails sampled, the lower third was preferred 46% of the time. Melampus coffea's preferred vegetation type is Spartina, and their second choice is saltwort. It's a succulent plant. Both days the coffee bean snails were spotted were on high tide days at 80 and 81 degrees. Literaria angulifera prefers the upper third of black mangrove trees. I found that mangrove leaves were more preferred than mangrove stems. This may be because leaves are an easier food source, with access to green organic matter. Mangrove periwinkles preferred mangroves 47.7% of the time. However, they preferred Spartina nearly as much, 45.6% of the time. This is the least vegetation-specific species. Literaria aerata prefer the upper blade of Spartina 42% of the time, and the middle third 37% of the time. Spartina periwinkles preferred Spartina 84% of the time, and their second choice was black mangrove leaves, but only 7% of the time. Polygyra cereolus preferred the lower third 78% of the time. They preferred water 64% of the time, more than any other marsh snail, and their second choice was Spartina at 21% of the time. This is a chi-squared test between vertical location and type of snail. Since the p-value is less than 0.5, that indicates association between vertical location and type of snail, which verifies what we saw in the bar graph. Literaria angulifera prefers black mangroves, and Melampus coffea and Literaria aerata prefers Spartina, which you can see in the circle graph. Gradulating creates wounds on live Spartina. This kills more biomass than is consumed by snails and slows plant growth. Periwinkles eat less than 37% of Spartina, but they damage the whole stem. Snails have a top-down control on Spartina biomass production and keep species in check. It is possible snails cause a microbial infection in Spartina. Snails graze on living Spartina even when dead Spartina is available. They create and maintain wounds with their radulas and consume dying tissues instead of green or dead tissues. In this study, there were 6 trials: resident snails, removal of snails, and addition of snails with resident snails. And all those trials were done again with fertilization, and it was found that radulations per stem nearly doubled when the marsh was fertilized. That shows snails are capable of regulating Spartina growth and feed vigorously when the plants are doing well in high nitrogen environments. This is important because it tells the importance of marsh snails in regulating Spartina growth. Now let's get into abundances. Literaria angulifera had the highest abundance at 51%. Literaria aerata had the second highest abundance of 36%. Melampus coffea had a species abundance of 12. Polygyra cereolus had an abundance of only 6. The overall preferred vegetation was Spartina. This is because Literaria angulifera and Literaria aerata are the 2 largest groups of snails, and they can both be found on Spartina. The second largest preferred vegetation was black mangrove leaf. That is because it is the top choice of Literaria angulifera and the most abundant species in my sample. I found that the upper and middle thirds are the most preferred vertical location. The most abundant species, Literaria angulifera, prefers the upper third. The second most abundant, Literaria aerata, prefers the upper third only 5% more than the middle third. Because these 2 snails dominate in preference for upper and middle third and have high abundance, their general vertical location preferences outshine other species and other locations. I found that some Literaria aerata species were living on coquina. I looked it up and it said that snails can eat algae and detritus that builds up in the pores of rocks. Adult Literaria aerata climb and forage at low tide and go into the sediment. When foraging in the sediment, snails tended to stay about a centimeter away from Spartina. In high tides, they climb to upper portions in the cord grass. Juvenile snails stay closer to the water than adults do to forage. This is because the base of the plant is not associated with temperature and desiccation stress. I saw a trend for juveniles in the lower third. However, I counted coffee bean snails as juveniles, and this may have skewed my results, because coffee bean snails are always small and always prefer the lower third. At high tide, snails climb up and avoid predators. In a study, Literaria aerata snails were caged in lower and upper portions of Spartina in North Florida. In general, snails caged lower to the mud had higher mortality and injury rates. In warm weather, snails forage in the substrate. The 29th was my warmest sampling day, and there was no increase in marsh snails in the lower third. High tide sampling days had increased presence of Melampus coffea and Polygyra cereolus. Spartina periwinkles preferred the upper third despite the tide. Another chi-squared test of independence between tide and vertical location of Literaria angulifera. Since the p-value is larger than 0.5, the null is true. There is no association between tide and vertical location. The tide does not affect Literaria angulifera's vertical location. This is consistent with my observations because mangrove periwinkles migrated from their leaves the least. Another chi-squared test of independence between Literaria aorata and tide. Since the p-value is less than 0.05, there is an association between the tide and location of Literaria aorata. On low tide sampling days, more of Literaria aorata was located in the upper third. I found that on rainy sampling days, the 22nd and 27th, snails were sticking to one another. I wrote this down and found that it could be breeding behavior, wintering behavior, or just movement. Snails may also just be more active on rainy days because there's less threat of desiccation. I noted 11 Literaria aorata and Literaria angulifera pairs, which may be evidence of hybridization between the two species. I expected that surface preference was going to be less specific on rainy and low tide days, but it wasn't. My graphs indicate no differences between surface preference and moisture. Literaria aorata keeps Spartina in check, but overgrazing from Literaria angulifera may cause even greater stress on Spartina health, causing mangroves to take over. Mangrove overgrowth may shade out Spartina. Either way, Spartina is so important in the natural manatee route because manatees stop at the St. Augustine water treatment and feed on marsh grasses and algae in the 312 salt marsh. Climate change can have huge overarching ecological effects, so reducing your carbon emissions can help save Spartina, snails, and manatees. So we learned that salt marsh snails have preferences in habitat, vegetation, and vertical migration. We found that Literaria angulifera was the most abundant and least specific. Polygyra cereolis, Melampus coffea were spotted in high tide sampling days. Literaria angulifera did not vertically migrate with the tides, but Literaria aorata did. Mangrove periwinkles surviving and thriving on Spartina and climate change-induced range expansion might allow them to outcompete Spartina periwinkles. As mangrove periwinkles dominate salt marshes, hybridization may be necessary in order for Literaria aorata species to survive. Big thank you to all the professors that helped me.
These are my citations, and thanks for watching.